Bismillahirrahmanirrahim. In this video, we are going to discuss the basement membrane composition and various molecules that are integrated with the basement membrane. Basement membrane is also termed as basal lamina. It is a thin, well-defined layer of specialized extracellular matrix. It separates the cell that synthesizes it from adjacent interstitial connective tissue. It has a thin, specialized extracellular matrix layer that separates epithelial muscles or nerve cells from the underlying connective tissue. It provides support, structure, and signaling cues essential for tissue organization and repair. So it is a very thin, visible as a fine line on microscopy. It is stained by periodic acid Schiff reagent stain, and this staining property is due to presence of glycoprotein. Its composition is type 4 collagen, laminin, nitrogen, and heparan sulfate proteoglycans. The key function of this basement membrane, it provides cell support and anchorage. So it's a mechanical support for epithelial tissue. The basement membrane also maintain the orientation of basal versus the apical domain. So it is important for the cell polarity. It transmits signals to stem cell and progenitor cells. So it is important for cell differentiation. It guides proper alignment of the cell and tissue. So it's one of the function is tissue organization. It is basically a scaffold for re-epithelialization. So its another function is regeneration and healing, and it is a crucial part of the neuromuscular junction. So it has also got a neuromuscular role. Where is it located? So it is located beneath the epithelial cells of the mucosa, skin, and glands. The surrounding muscle fiber, smooth and skeletal muscle. In peripheral nerve, it ensheathes the Schwann cells, enveloping the Schwann cells. It encases the adipocytes around capillaries between endothelium and pericytes, and it is present in supporting ducts and tubules in organs such as kidney, salivary gland, etc. On microscopy, it is a PS positive structure that is very thin, and it is rich in glycoprotein, and it is best visualized in electron microscopy, showing two definite or distinctive structure, lamina lucida and lamina densa. One is densely packed area and another is lightly packed area. So this is a picture scanning electron micrograph of basement membrane. Over here in this picture you can see that these epithelial cells are lying on this basement membrane. This is actually a basement section from chick embryo corneal epithelial cells, and these epithelial cells are distinguished from underlying connective tissue. The core component of the basement membrane are collagen type 4, laminin, intactin or nitrogen, and perlecan. That is the heparan sulfate proteoglycan. So the collagen type 4 and laminin, they are present in isoforms, and these many isoforms are responsible for its tissue function and tissue type. So these collagen type 4 are a structural protein, and they form a covalently cross-linked lattice for tensile strength, while the laminin is a glycoprotein that establishes initial scaffold and mediates cell adhesion and polarity. Intactin, that is also known as nidogen, it is a connector protein that links collagen 4 and laminin network. And perlecan at charge selectivity, it is a large heparan sulfate proteoglycan. It at charge selectivity binds with growth factor and various chemokines. So all these together form the basement membrane. Laminin network is non-covalent at the base. Collagen 4 is covalently bound, and it acts as an overlay. It is formed as an overlay at the base of the laminin, and the these are bridged by nitrogen and perlecan. So these isoforms of collagen 4 and laminin, they are responsible for the tissue specific signaling, developmental cues, and diverse cell to matrix interaction. The basement membrane is not just a mat under your skin. It's a dynamic signaling platform, molecular reservoir, and developmental guide. It has got multiple function. So whenever somebody asks what are the function of the basement membrane, it supports the cell differentiation by providing biochemical and mechanical cues. It's a barrier and filter, especially in kidney glomeruli and capillaries. It provides a migration surface for epithelial cells post-injury during reepithelialization. So it directs the reepithelialization process. Then it is an anchoring platform by stabilizing epithelial and endothelial cell layers. It is also has also got function in neuromuscular repair because it reforms neuromuscular junction after nerve injury. It guides tissue patterning during embryogenesis, so it has got importance in developmental morphogenesis. It has got another important function, that is growth factor reservoir. It stores and present many growth factors such as fibroblast growth factor, VEGF, transforming growth factor, chemokines, and peptides, and it releases and provides at the time of the need. It also determines cell shape and polarity by organizing cytoskeleton through integrin signaling. Now first we will see the constituents of the basement membrane individually, and then we will finally see its organization, how it is these elements are forming the basement membrane. So perlecan or heparan sulfate proteoglycan has got one protein core and three heparan sulfate, and there are glycosaminoglycan chains, and it is associated with laminin, collagen 4, fibronectin, growth factors, and chemokines. The laminin has got many chains, alpha, beta, and gamma chains, and there are 16 isoforms of these laminin that are tissue specific and function organ specific. They are heterotrimers with alpha, beta, and gamma chains. There are 5 types of alpha chains, 3 beta, and 3 gamma chains identified. So this is its molecular structure. It is a cross-like structure basically, and these alpha, beta, and gamma are forming this cross shape appearance of the laminin molecule. So the association of the molecule with this laminin are integrin, dystroglycan, and other receptor on the variety of the endothelial, epithelial, muscle, Schwann, and adipocytes. They form self-associated non-covalent network that organizes basement membrane. The other molecular association are other lamellin. They are bind with the other lamellin, nadogen, intactin, perlecan, agrin, and fibulin. Now the nadogen or intactin is a two member family monomeric molecule, and it basically anchors the laminin and other structure. So the molecular association are collagen 4, fibulin, perlecan, laminin, and it stabilizes uh, basement membrane by associating itself with the laminin and collagen type 4 network. So it keep them together. It is basically connecting these two, collagen 4 and laminin. So collagen type 4, the major and important uh, part of the basement membrane, is more than 3-member family heterotrymic chain, and it is selected from 2 or 3 of 6 unique alpha chains. 3 single chains from alpha helical tail of collagenous region and association of 3 globular region. So this molecular structure, it forms an overlay, and it this this is important for the various function. This overlay is important for the various function that is performed by the basement membrane. There is molecular association by integrin receptor of many cell and form covalent association. The other one, lamellin, was non-covalently attached, and here these have got covalent association, and they are self-associated, and they also associate with collagen 4, perlecan, nitrogen, intactin, and spark, that is secreted protein, acidic and rich in cysteine. So this illustration is describing the organization of all these elements and how they are forming the basement membrane. So these integrin receptors that are over here in the plasma membrane, these integrin receptor, they are bound with the laminin, and the laminin uh, is also down there. It is this overlay. This laminin is in connection with the overlay of the collagen type 4, and these molecules that are perlecans composed of glycosaminoglycans and protein core and heparan sulfate, these perlecan, 3 heparan sulfate, protein core, and glycosaminoglycan, they are also attached with the laminin. Okay, and These are the bridging nitrogen or intactin. They are holding these laminin with the collagen fiber. So here this is forming these integrin receptors are attached with the laminin, and down there this overlay of the collagen type 4 is providing the support, while the intactin or nitrogen, they are providing the bridging among these fibers. So this schematic diagram is showing the typical component that are found in the basement membrane, and a basal keratinocyte over here is adhered to the underlying basement membrane and dermis via the focal adhesions that transmit mechanical force, and regulatory signals consist of numerous interacting components such as hemidosmosomes and other uh, antigens. So there are receptors for laminin on these cell membrane. These are integrin, they bind laminin laminin, and there is perlecan, that is the large molecule of heparan sulfate, large molecule. So there is presence of these nitrogens or intactin that are connecting perlecan with the laminin, and hence they are attaching these epithelial cells to the underlying collagen, that is type 1 and type 3, with the help of anchoring fibrils, where there are many anchoring fibrils also present. Thank you for watching this video. If you have any query, please write in the comment box.